Hello, good day to all medical technology students out there. Here I am, your one and only Miss Merle, to do the discussion for the complement system. Well, what is the complement system? We should remember that the complement system is a crucial mediator of the innate immune response, contributing to cell homeostasis, tissue development and repair, reproduction, and crosstalk with other endogenous systems. Each of the three major complement activation pathways, the classical, the alternative, and the mannose binding lectin pathway, employ specific recognition molecules and initiating serine proteases. All the three pathways converge into a common sequence. So here. This leads to the formation of the biologically highly active anaphylatoxin C5a and the C5b through 9 membrane attack complex, the MAC. This one. The latter forms transmembrane channels. The MAC is responsible for creating pores or channels, which either induces sublytic activation of the cell or results in target cell lysis. Primarily as an important branch of the first line of defense, the complement protects the host from invading pathogens and abnormal self-derived components. On the other hand, uncontrolled complement activation may lead to life-threatening effects such as systemic inflammation and shock, dysregulation of the coagulation and fibrinolysis or fibrinolytic pathway, and also autoaggression. Furthermore, under certain conditions, the regulated complement activation may also promote tumorigenesis. So for the details that explain the hows and the whys of things, let's get started in the next slide with the outline. So this time we will cover the following. So we will define what complement is, what is the complement cascade system, what are the biological or physiological functions of the complement, the nomenclature of the complement system, the pathways. The most important topic will be the complement activation pathways, which much of our talk will be centered, and we have complement production or regulation or regulatory system controls. We also have the terminal membrane attack complex or MAC, and we will also be talking about disease states in correlation to complement, specifically the complement deficiencies, and lastly we will have the laboratory detection of complement abnormalities. So let's start. Whenever a pathogen breaches the host epithelial barriers and initial antimicrobial defenses, it next encounters a major component of innate immunity known as the complement system. Or complement. And what is it? Well, complement is a collection of soluble proteins. They are soluble proteins present where in our blood, and other body fluids. It was discovered in the 1890s by Jules Bordet. So it was Jules Bordet who discovered it first. Complement as a heat labile substance in normal plasma whose activity could complement the bactericidal activity of immune sera. So that was it. Although complement was first discovered as an effector arm of the antibody response, we now understand that it originally evolved as part of the innate immune system, that it still provides protection early in infection in the absence of antibodies, though more ancient pathways of complement activation. The complement system is composed of more than 30, take note, more than 30 different plasma proteins. So more than 30 different plasma proteins, which are synthesized mainly by the what body organ? The liver, so that you will not forget, there's a letter L in the word complement. Think of that letter representing the liver, in which majority or mainly much of the plasma proteins are produced. The complement proteins are produced. In the absence, you might think, is the complement cascade or system always turned on? Is it turned on constitutively? No. In the absence of infection, these proteins circulate in an inactive form. So, in absence of infection, complement proteins are inactive. Inactive. Take note. No infection, complement proteins are inactive. But in the presence of pathogens or antibodies bound to pathogens, the complement system becomes activated. So that's the time that there is activation again in the presence of pathogens or antibody bound to pathogens. Well, particular complement proteins interact with each other to form several different pathways of complement activation, all of which have the final outcome of killing the pathogen either directly or by facilitating its phagocytosis and inducing inflammatory responses that help fight the infection. So that's it. And now we have the biological functions or the physiological functions of the complement. Which fall into the following two general categories. Well, we have the cell lysis by the membrane attack complex and the biological effects of proteolytic fragments of the complement. So again, we have cellular lysis or cell lysis and biological effects of proteolytic fragments of the complement. So most often, majority of the books would say that the endpoint of complement activation will always be cellular lysis. So let's talk about the first category. This one. The first category is the situation in which the MAC leads to osmotic lysis of a cell, while the second category encompasses other effects of complement in immunity and inflammation that are mediated by the proteolytic fragments generated or made during the activation of complement. And these fragments may remain bound to the same cell surfaces at which complement has been activated or may be released. So remember, fragments may either be remain bound or released into blood or extracellular fluid. Well, in either situation, active fragments mediate their effects by binding to specific receptors expressed on various cell types of cells, including phagocytic leukocytes and the endothelium. Well, in contrast, the absence of integral component of the classic, alternative, or terminal lytic pathways can lead to a decreased complement activation and a lack of complement-mediated biological functions. So, here this is the function these sorry are the functions of the complement as told by Reut. So we have the complement can trigger and amplify inflammations or inflammatory reactions, and can also our complement can also of course is functions in chemotaxis, can attract phagocytes and enhance chemotaxis. We also have the clearance of immune complexes and apoptotic cells, cellular activation for microbial killing or could be direct microbial killing, and the complement also plays roles in the development of antibody responses, which we can say highly efficient in nature. So the immune defense and pathological effects of complement activation are mediated by the fragments and complexes generated during activation. So those fragments, those complexes are generated upon activation of the complement. So the small pro-inflammatory fragments, they are small because they are having the A, like for example C3A and C5B. Later in the nomenclature I will explain that. The small pro-inflammatory fragments C3A, C3A and C5B, they are responsible for the chemotactic activity of the complement. So if we are talking about chemotaxis, what fragments or complement fragments shall we blame? So not blame, but who are responsible for it? These fragments C3A and C5B. While for we have the large fragments too, the C3B and the C4b. These are the large fragments, C3b and C4b. They carry opsonization. So if we have chemotaxis, and for chemotaxis we have C3A and C5b. For opsonization we have C3b and C4b. However, if we are talking about how these antigen antibody immune complexes are being cleared or disposed away from the tissues, we have C1q, and the bonded fragments of C3 and C4. So C1q and C3 and C4. For the disposal of immune complexes from the tissues are brought about by C1q and equivalently bonded fragments of C3 and C4. Moreover, the lysis after this killing, surely it will lead to lysis. These are microbial killing were secondary to lysis. So lysis of mammalian and bacterial cells are done by the MAC, or the membrane attack complex, from C5b through 9. Again, for chemotaxis, the complement of fragments responsible are C3A and C5b. For opsonization, the large fragments C3b and C4b, while those for the clearance or disposal of antigen antibody immune complexes, we have C1q and the covalently bonded fragments of C3 and C4, while for the lysis or the cellular lysis happening at the end point, at the end of each complement activation, we have MAC responsible for it. MAC, or membrane attack complex, starting from C5b through 9. So C5b, 6, 7, 8, 9. Okay. This one is another cartoonized table representing the functions of the complement in a direct and simplified approach. So for this one, row A, the major functions of the complement system in host defenses are shown. So we have in row A, cell-bound C3b, cell-bound C3b is an opsonin that promotes phagocytosis. So C3b promotes phagocytosis of coated cells, while row B, the proteolytic product C5A, proteolytic product C5A, C3A, and C4B to a lesser extent stimulate leukocyte recruitment, or the recruitment and activation of leukocytes, so that they can have an effect on inflammation and eventually leads to the destruction of microbes by leukocytes. And row C is representing row C here represents how the MAC, the membrane attack complex, brings forth lysis to the cells. So we have starting this one, and then once C9, and then it creates a pore in the cell membrane, and then it leads to cellular lysis. In this one, these tabulated physiological functions of the complement as presented by Turgeon is somehow categorized into 3 pertinent activities. We have a host defense against infection. We have interface between innate and adaptive immunity, and the disposal of waste. Somehow the same thing, but they are just categorized in different ways or said in different ways, but main themes run through. So under host defense against infections, we have opsonization, as what I have said earlier. The covalently bonded fragments of C3 and C4 is just the same. We have the ones responsible for chemotaxis, C5A, C3A, and C4A, they are anaphylatoxins, and the lysis brought about by MAC. However, as what I have said in the previous slides, that complement can also augment or plays roles in the efficient antibody immune responses. So augmentation of antibody effects, we have it's being brought about by C3b and C4b, which are bound to the immune complexes and to the antigen. We also have enhancement of immunologic memory, and also lastly, for the disposal of waste, the clearance of immune complexes C1q and C3 and C4, and also those cells that are already senescent and about to die. It's also the role of the complement. So, well, the complement has a really a central role in inflammation. Think of a complement. If you have an inflammation, the complement is acting there because it causing chemotaxis of phagocytes, activation of the mast cells and phagocytes, and lastly, the opsonization and lysis of pathogens, and the clearance of these antibody immune complexes. So for the complement nomenclature, for the complement proteins can seem confusing. So we will start by explaining their names. The first proteins discovered belong to the classical pathway, and they are designated by the letter C followed by a number. So, example, the native complement proteins such as the inactive zymogens have a simple number designation. For example, we have C1 and C2. Unfortunately, we should remember that they were named in the order of their discovery rather than the sequence of reactions. Again, unfortunately, they were named in the order of discovery rather than the sequence of reactions. The reaction sequence in the classical pathway, for example, it's not like C1, 2, 3, 4, 5. No, it's C1, C4, C2, C3. Take note here. However, starting C5, it's already okay. 5, 6, 7, 8, and 9. So again, complement proteins are named in the order of their discovery and not based on the sequence of their reactions. Okay, are we clear on that? So, what else? We also have the cleavage reaction products. They are designated by adding a lowercase letter and a suffix. So, example, the cleavage of C3 produces C3A, a letter, adding a lowercase letter and a suffix. Lowercase letter and a suffix. So, for the small fragments, A, larger fragments, B. See? The larger fragment for other factors is designated by the suffix B. So, if the fragment is large, suffix is B. If the fragment is small, the suffix will be A. However, there are exceptions. For C2, the larger fragment was named C2A by its discoverer. See, larger, but it's A, it's not B. Also, another exception will be the naming of C1q, C1r, and C1s. These are not cleavage products of C1 but are distinct proteins that together comprise C1. And other exceptions, the proteins of the alternative pathway were discovered later and are designated by different capital letters. For example, factor B and factor D. So we cannot, we are not having, if we the complement proteins in the alternative pathway, we will call them factors, not as complement proteins, okay? And their cleavage products are also designated by the addition of lowercase a and b. Thus, the large fragment of B is called BB. Thus, the large fragment of B is called BB and the small fragment BA. Again, cleavage products are designated by the addition of lowercase a and b. If the fragment is large, the large fragment, so it has to be, it's called BB, and the small fragments, still it follows A. Activated complement components are sometimes designated by a horizontal line. For example, this one, it's supposed to have this horizontal line here. But it's not commonly used in textbooks. I've only seen this one in Turgid. Next, we have the complement activation pathways, the classical, pathway, the alternative, and the lectin pathway. And we should again remember that one major function of complement is to label pathogens. See, labeling pathogens for recognition and other foreign or toxic bodies for elimination from the host. The complement activation pathways have evolved to serve this purpose, and the multiple ways in which activation can be triggered together with intrinsic amplification mechanisms ensure efficient recognition and clearance. So the labeling process allows or permits an efficient recognition and clearance of such pathogens or foreign or toxic bodies. Moreover, there are several different pathways to activate the complement system, so providing a large degree of flexibility in response. The first activation pathway to be discovered, yes, was now termed the classical pathway. So this was the first pathway to be discovered. It is initiated by antibodies bound to the surface of the target. Although an efficient means of activation, it requires an adaptive immune response. That is, what the host must have previously encountered. The host must have previously encountered the target microorganisms in order for an antibody response to be generated. While the alternative pathway described way back in 1950s, the alternative pathway provides an antibody independent, so it doesn't require antibodies. If we have the classical pathway dependent on antibodies, the alternative and also the lectin pathway are antibody independent mechanisms for complement activation and pathogen surfaces, while the lectin pathway is the most recently described activation pathway. So if we will, the first to be discovered was the classical, followed by the alternative pathway and the lectin pathway. Again, the lectin pathway, it is the most recently described activation pathway, and it also bypasses antibody to enable efficient activation on pathogens. What do you mean by that? That again, the alternative and the lectin pathway are both antibody independent pathways. It doesn't require the presence of antibodies, while for the classical it requires antibodies for the complement to be activated on the pathogenic surfaces. So if we will try to summarize all these pathways, like what are they, like put them into words, these are the stages. There are common themes in them, especially in the phases of each pathway in eliminating a microbe. In fact, yeah, proteins that can distinguish a cell from microbial surfaces in the yellow box, activate a proteolytic amplification cascade that ends in the formation of a critical enzymatic activity, the green box. Activity of what? The critical enzymatic activity of the C3 convertase, a family of proteases, and the C3 convertase. This activity is the gateway to three effector arms of complement that produce inflammation, and enhance phagocytosis. And the enhancement of phagocytosis, for a while, my laptop is getting low on battery. Okay, again. So it enhances phagocytosis, from inflammation to phagocytosis, and also lyse microbial membranes. So the membrane attack leads to lysis. So if I will ask you what are the stages of complement action or activation. We have the following: pattern recognition trigger starts with the labeling, and then the proteolytic cascade brought about by C3 convertase, started by C3 convertase, followed by inflammation, phagocytosis, and the membrane attack complex, which leads to cellular lysis. So it's here: phagocytosis, the lysis, and inflammation. So it's easy to remember. The pattern recognition triggers in yellow, and then the protease cascade amplification here, and we have inflammation in purple, phagocytosis, and the membrane attack. I'm sorry I write a lot. That's the way how I study too. So all three pathways, classical, alternative, and lectin pathways, involve the activation of C3. So what is common to all three pathways? Common, we have two things in common: the involvement of the C3 activation, and also there is this it comprises a proteolytic cascade in which complexes of complement proteins create enzymes. So again, the involvement of the activation of C3. C3 is the most abundant and most important of the complement proteins. Again, C3 is the most abundant, most important of the complement proteins. And then the second one is about the proteolytic cascade, in which complexes of complement proteins create enzymes that cleave other complement proteins in an ordered manner to create new enzymes, thereby amplifying and perpetuating the activation cascade. And then each of the activation pathways generate a C3 convertase, which converts C3 to C3b. That C3 convertase, how to say, is responsible for converting C3 to C3b. C3 becomes C3b in the presence of that C3 convertase. And this event here, is the central event of the complement pathway. C3b in turn activates the terminal lytic membrane attack pathway. So C3b, it's responsible for the activation of the MAC. So who activates the MAC? What fragment? It's C3b if this is a person. So I'm using who. And then the first stage in the classical pathway is the binding of an antigen to an antibody. So again, antibody-dependent classical pathway. The alternative pathway does not require antibody and is initiated by the covalent binding of C3b to hydroxyl and amine groups on the surface of various microorganisms, while the lectin pathway is also triggered by microorganisms in the absence of antibody, with sugar residues on the pathogen surface providing the binding sites. And the alternative and lectin pathways provide antibody. Again, I would just like to note the alternative and the lectin pathways provide antibody-independent innate immunity, whereas the classical pathway represents a more recently evolved link to the adaptive. So if you will ask me, the complement system functions in innate immune response, or only in the adaptive immune response, I would say both, because for the antibody-independent innate immunity, it's being brought about by two pathways, the lectin and the alternative pathway, while the antibody-dependent adaptive immune responses are brought about by the classical pathway. So again, if I will ask you, complement system functions only in the innate immunity, wrong, because the complement system can function in both. Regarding specifically, the answer lies if I am talking about what specific pathway. Again, for classical innate, is it innate? No. Innate, we have your what? The alternative and the lectin pathway. while for the adaptive immune responses, it's the classical pathway. Next, here. So, how are the things going on in each pathway? So let's start. The pathogen recognition mechanisms of the three complement activation pathways are shown in the top row here. In the top row, along with the complement components used in the proteolytic cascades leading to the formation of a C3 convertase. In the middle row, this one, this enzyme activity cleaves complement component C3 into small soluble protein C3A and the larger component C3B, which becomes covalently bound to the pathogen surface. The lectin pathway of complement activation here, top left, lectin pathway is triggered by the binding of mannose binding lectin or ficolins. Mannose binding lectin or ficolins to carbohydrate residues in microbial cell walls and capsules. So this is the microbial surface or the pathogen surface. Okay, again, the lectin pathway is triggered by the binding of MBL, mannose binding lectin, is a lectin that will bind to the mannose residues present in the pathogen surface. That's it. And or ficolins to carbohydrate residues in microbial cell walls and capsules. While in the top center, we have the classical pathway. Classical pathway is triggered by binding of C1 either to the pathogen surface or to the antibody bound to the pathogen. Miss, where is C1? Because C1 is composed of a complex of C1Q, C1R, and C1S. So this is C1, the C1 complex. Because it's a complex of three subunits, C1Q. Okay, I'm sorry, my laptop turned off. I don't know why. So let's go back again. We have the three pathways: classical, alternative, and the lectin pathway. And I am talking previously about how these the activation, what are the triggers for each pathway. So let's start again. I will go back. Sorry. So we have the pathogen recognition mechanisms and how they recognize pathogens. Again, for the how to say, we have the sorry.